speaker of the session, kind of is going to go down to more of a species level and how the conservation lessons learned. And that's Dr. Kerry Byrne, and she's a professor with the Department of Environmental Science and Management at Humboldt State University. She has a BS in Environmental Biology and Management from UC Davis and a PhD in Ecology from Colorado State University. Dr. Byrne is an applied terrestrial ecologist with research interests in rare species conservation and management, the effects of anthropogenic climate change on plant community composition and structure, and terrestrial restoration. So please, and the title of the talk is Restoring a Population of Applegate's Milkvetch, Lessons Learned for Conservation. Welcome.
Thanks, Claire, for that introduction, and thanks for inviting me to give this talk. I wanted to start out with this photo. This is, believe it or not, this is one of the, well, it is the largest remaining population of Applegate's milkvetch in the world. Well, at least part of it. You can't see the whole population. And this is at the Klamath Falls Regional Airport. So not a very pristine environment, which is why I wanted to include the photo. Although I like to call it the golden hour from about 6:30 a.m. to about 8:45 a.m. Before the F-15 started taking off. It's also a National Guard base, so there'd be planes taking off all the time. You can't really see it, but right on the other side of this fence right here. All right, so I'll get back to this photo later in my talk. So what I'm going to talk about today is first give a brief introduction to the Astragalus genus. Then I'll talk a little bit about the conservation story of Astragalus applegatei, or Applegate's milkvetch. I'll give a brief overview of the research I'm doing with undergraduates and graduate students at Humboldt State and my previous institution, Oregon Institute of Technology in Klamath Falls, Oregon. And then finally, I want to tie this together and talk about some more general lessons learned about for conservation based on the research that myself and other people have been doing over the years. We'll start with an introduction to the genus. So I wanted to first, before I started talking about Astragalus, I wanted to see a show of hands how many of you have actually worked with any Astragalus species in your work as a botanist. Probably, I'm looking across the audience, maybe, I don't know, maybe 90% of you have. And that's not super surprising because it's the largest flowering plant genus in the Fabaceae. There's over 3,200 species described worldwide, and within California, there are around 150 taxa, over half of which are considered rare, threatened, or endangered by the California Native Plant Society. And there's a lot of different reasons for this. And some of them are the genus has really diverse abiotic and biotic requirements. For example, there's a lot of soil specialists within the genus, a lot of narrow endemic taxa. And as an example of some of the various or diverse biotic requirements, they have really diverse reproductive strategies, ranging from obligate outcrossing to cleistogamous selfing. And a 2011 paper just said that the reproductive biology of only 29 species has been published so far. So out of the 3,200 species worldwide, we really don't know about the reproductive biology of that many different species. So there's a lot of research still to be done on the genus, and particularly about rare plants within the genus. So if you want to kill half a day, or if I'm honest with myself, maybe longer, what you can do is search on CalPhotos for Astragalus. So this shows you some of the diverse Astragalus species that you can find within the state of California. You can see some of the different growth forms, floral colors, sizes, and soil or different edaphic requirements of the species within the state. And in this next slide, you can see some of the different reproductive parts, or legumes. So it's a really diverse genus, and we all know this because most of us have worked with Astragalus species. So next I wanted to talk a little bit about the conservation story of Astragalus applegatei. This species is not actually found in California. I'm sorry to disappoint you all. It's found just over the border in Klamath Falls, Oregon. Yes, there's a little chair in the back of the room. So I lived there for about 4.5 years, and that's when I became involved with the species. But a little background, it was first described in 1936. It was believed to be extinct until 1983, and then was listed as endangered federally endangered in 1993, and has been on the endangered species list since then. It's also an Oregon state listed species as well. It is currently known, well, it's the only remaining populations are near or within the city of Klamath Falls, Oregon. So it's a bit of a challenge because when you're working with this species, you're working in very developed areas. Here we can see this is the Klamath Falls Airport, and this map shows basically the city of Klamath Falls and 3 of the major population sites. Only 2 of the major population sites are actually protected. The total population estimate is around 30,000 individuals for the species. And I included this final point, the fish and wildlife biologist who was charged with the species recovery. His name was Ron Larson. He's since retired. In case you didn't know, in Klamath Falls, Oregon, the main issues are endangered fish and water. And so in the Fish and Wildlife Service office in Klamath Falls, there's not any botanists. But it was kind of a stroke of luck that Ron Larson, who's actually a marine biologist, was charged with the recovery of this species because he was really instrumental in getting funding, and he had a lot of passion, or he still has a lot of passion for the species. So that was a wonderful stroke of luck, even if he's not actually a botanist. So here on the right, you can see Applegate's milkvetch in a photo that I took. It's just a low-growing perennial forb with mostly lavender flowers, and it can have stems ranging from 1 to, I think some of the largest plants have over 200 stems. The size really varies depending on where you are, if you're close to water or not. Like all Astragalus species, it's a taprooted plant, and so transplant of adult individuals isn't possible. So that can be a real challenge when you're trying to, when any sort of construction is happening, or for example at the Klamath Falls Airport, you can't transplant plants that are being killed when there's any sort of construction that happens. And finally, it has really challenging germination requirements. Like most Astragalus species, it has a really hard seed coat, and so the seeds must be scarified in order for germination to occur. We don't know it does this in the wild, but we found that sandpaper or 29-gauge hypodermic needles work really well to puncture the seed coat. But more interesting are the microbial symbionts that it has, and that's what I'm going to spend a little bit of time talking about. So all species within the Fabaceae have a symbiotic relationship with rhizobium bacteria. You can see the little root nodules here in this photo. So that's where nitrogen fixation occurs, and so Astragalus applegatei also has those root nodules in a relationship with rhizobium bacteria. But what was not known, what's unique about this species, is the relationship with arbuscular mycorrhizal fungi within the genus Calamus. And this is fairly recent knowledge. When the species became threatened and endangered, there was a lot of work to try and grow seedlings for transplant into existing populations to grow the population numbers. And so researchers from Oregon State were able to successfully germinate seeds, and then they grow until they were somewhere between 6 and 12 weeks old, and then the seedlings would just die in the greenhouse. They didn't really know what was going on at first. Next, they tried growing seedlings with commercially available inoculants, and the same thing would happen. Somewhere between 6 and 12 weeks, those seedlings would die. So finally, just sort of by trial and error, they decided to dig up some soil from existing native plants, mix it in with the potting soil mix, and then grow those seedlings. And that was the first time those researchers were actually able to get those seedlings to survive past about 12 weeks old. So that was a huge success in terms of conservation for the species. And then in the late 90s, this paper was published. They were able to identify the genus of the fungi, but they weren't able to, no one knows if it's a single species, if it's a group of species, or if it's a new species to science. So actually one of my master's students, that's part of her work, is to do some additional follow-up work to figure out more about this mycorrhizal fungi symbiont. All right, so I did also, in addition to what my graduate student is doing, I wanted to talk a little more about the ongoing work that I have going on in Klamath Falls, or back on campus. So I have 3 main projects that are going on, and I'll just very briefly describe the 3 of them. The first is to determine the longer-term survival of transplanted seedlings. So once the Fish and Wildlife Service was able to grow, once the mycorrhizal fungi was identified and the seedlings were able to be grown and cultivated in greenhouses, the Fish and Wildlife Service hired a local native plant nursery to start growing seedlings for transplant into an existing population at a very small Nature Conservancy preserve in Klamath Falls. And this is the Nature Conservancy preserve. It's called Uwana Flats Preserve. You can see just sort of some industrial activity going on in the background. It's not in a pristine environment, but there are existing native individuals there, and so the Fish and Wildlife Service paid this local native plant nursery to raise seedlings for 3 years, 2012 through 2014, and then I worked with students from Oregon Institute of Technology to transplant those seedlings into existing populations. So we marked all of those seedlings with unique number tags and are monitoring the success of those transplant efforts over time. The second project that I have going on is looking across different populations of the species and doing some demographic data collection, some monitoring, and we'll use the data that we're collecting to create a stage-based population model of the species to hopefully make some predictions about what's going to happen with the species more long-term. And then third, I worked with an undergraduate from Oregon Institute of Technology to do a study on the reproductive biology of the species. And so she did a pollinator exclusion experiment at Uwana Flat Preserve and some other sites as well. So today I'm just going to talk about the results of the first and the third projects. So in the transplant project at Uwana Flat Preserve, which is, I didn't mention this before, it's kind of like a postage stamp preserve. It's a little under 7 acres, so it's pretty small. But over 3 years, over 1,300 seedlings were transplanted at this Nature Conservancy preserve. And here you can see the year that each of the cohorts were planted and their survival rates. So the 2012 cohort has a 21% 5-year survival rate. The 2013 cohort has a lower, a 12% 4-year survival rate. And the 2014 cohort has a 20% 3-year survival rate. So these numbers may seem a little bit low, but actually they're pretty good, I would say. Particularly because we were not going out and watering the transplants. We just put them in the soil and then came back the next year or every summer to check on their survival. So I think a 20% survival rate is pretty decent for this species, given the climate and the harsh conditions in Klamath Falls. So we're pretty happy with these results. The second project I wanted to talk about was the Pollinator Exclusion project, which I did with an undergraduate at Oregon Tech. So basically what she did is she selected native occurring individuals within populations of Applegate's milkvetch. She put a mesh bag over one flowering stem, and that served as the pollinator exclusion. And then on the other stem, she left it open, so it was an open control. We don't know whether pollinators visited those flowers or if they were self-pollinated. We can't tell that for sure. All right, so I'm not going to spend a lot of time talking about this figure, but in blue we have the open pollinated control, and then yellow is where pollinators were excluded. And first of all, and on the x-axis we have different sites where she did the experiment, and we're looking at viable seeds produced per stem. So the first kind of take-home message is that viable seeds varied by site. And so our different populations had different seed production per stem. And then second, excluding pollinators reduced seed set in 2 of the 3 sites. So we need to do more research on this, but in the case of Applegate's milkvetch, it appears that pollinators are important in the seed production. All right, so finally, a few lessons that we learned from this work for conservation. Okay, I wanted to go back to this photo that I showed at the beginning, which is at the Klamath Falls Airport, a not very pristine site for rare plant conservation work. And so the first take-home message is, in a lot of cases, we need to take advantage of undeveloped pockets of lands in suburban or urban environments for species conservation. This is not an age where we can find pristine lands for conservation. So if it's a species that occurs in an urban environment, you gotta take advantage of where you can get the land and where the species is already. Second, and the microhysal research really showed this, that restoring plants to a community isn't enough. We need to think about the entire biotic and abiotic community in order to make restoration a success. We can't just plant an individual and expect it will survive. We need to think about the fungi, we need to think about the pollinators, etc. And so this is something that I'd like you all to, probably you all know this already, but this really illustrates that point, that we really need to think about the entire community. And lastly, getting the community excited about the project might be as important as the project itself. In the case of Applegate's milkvetch, this fish and wildlife biologist, Ron Larson, was really able to get the community involved. People who didn't have a background in botany. One man became the Nature Conservancy Preserve volunteer manager for that preserve. And he would go out, he still does, he goes out every week on his own time and checks on the preserve, makes sure that no one is leaving trash or anything like that. So that was really instrumental on Ron Larson's part. And then he also got me involved, which has been a great stroke of luck for me. I've gotten a lot of students involved. So if you have the opportunity to get students involved in your research or in your conservation work, I think it's really valuable. When I told my students that I was leaving Oregon Tech where I used to work, their first question was always, what's gonna happen to Applegate's milkvetch? So I think that's really important, and I just encourage you all to get other non-botanists involved in any sort of conservation work that you do. So with that, I'd like to thank some of the people that I've worked with and my funders over the years, and I'm happy to take any questions if you have any.